Okay, well, we got cut off a little bit on that last video right there. It was talking about movement of water and the two means by which water moves through the phospholipids and the aquaporins, and I was just saying that the aquaporins is probably the major means of moving water into or out of the cell. And these aquaporins, as pointed out previously, they are the channel proteins, essentially pipes, that allow very rapid movement of water across that plasma membrane. Okay, so let's go on over to this other topic now. And we're still in Chapter 5 for General Biology 1, and it's Section 4, so Section 5.4, which is passive movement of materials into and out of the cell. Another form of passive movement of materials into and out of the cell, besides the diffusion that we've already talked about and the osmosis that we talked about, is facilitated diffusion. Facilitated diffusion is a diffusion through a protein. So it is diffusion. It is movement down or along a concentration gradient. So it is a form of passive transport. The cell does not have to do any work to cause this to happen. And an example of facilitated diffusion occurs when glucose enters red blood cells. And so let's take a look at a diagram for that. And there we go. So right here we have a blood vessel. And here is a red blood cell, RBC, red blood cell. Now, out here in the plasma, blood plasma, we have an abundance of glucose out here. Remember, we maintain a concentration of about 1/10 of 1% glucose in the bloodstream. Now, here in the red blood cell, we're going to have far less glucose, because one of the things that happens as soon as this glucose enters the blood cell, it is going to be converted to glucose phosphate, and so it's kind of taken out of that. It doesn't really build up to the same level in the cell as what we have out there in the cytoplasm. So simple diffusion would say, well, this glucose ought to just keep sliding right on into that cell. But there's a little problem. The glucose molecule is too large just to go across those phospholipids. So we have to have a special protein right here that allows this glucose to pass through the protein. And the glucose is going from where it's in high concentration to where it is in a low concentration. So it is going downhill. It just has to go through a protein in order to move across that membrane. So this is still diffusion, and it is facilitated or made possible by the protein. So this is a passive process since it is going downhill. Anytime it's diffusion, it is a passive process. Okay, so let's go on over here and this. So the mechanics of facilitated diffusion. One protein that will move glucose into the cell is one called glucose permease. As glucose permease attaches to a glucose molecule on the outside of the cell, and that causes the glucose permease to change its shape. The glucose is then released to the inside of the cell. The glucose permease returns to its original shape, and it is ready to pick up another glucose molecule on the outside of the cell. So notice that this glucose permease is a carrier protein. The carrier protein is one that will bond with the material it's transporting, change its shape, drop the material off on the inside of the cell, go back to its original shape. So the channel protein is the one that acts like an open pipe. that may or may not be gated. And the movement through the channel proteins is much more rapid than the movement through the carrier proteins. Okay, so here's an example. And in this example, we're actually moving material to the outside of the cell, but you notice right here, this material is in much higher concentration inside the cell than out, and this is a material that cannot go through that phospholipid bilayer, so it has to go through the protein. So it's still diffusion, going from area of high concentration to low concentration, but it's going through a protein, so it is being facilitated by, or assisted by, or made possible by the protein. So it is facilitated diffusion. There is no requirement for energy from the cell to allow this to occur. Okay, so that was pretty short right there for facilitated diffusion, but the main thing to recognize, facilitated diffusion is still diffusion. It is movement of material downhill from area of high concentration to an area of low concentration. It's just that we're dealing with a material that is either a larger organic molecule and cannot go across the phospholipid bilayer because of its size, or it is an ion and cannot go across that phospholipid bilayer because of strong electrical charge. And those types of materials have to move across that plasma membrane by going through a protein. Now, that protein could be either a carrier protein or a channel protein, but it has to go through a protein. And since it is going downhill and there is no, it's going down or along the concentration gradient, there is no expenditure of energy on the part of the cell. So it is a passive process. Okay, thank you.